Hello, everybody. Good morning and good afternoon, and welcome to today's webinar, Automated One-Stop Purification Solution for Plasmid, Antibody, and Protein. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by GenScript. To learn more, visit genscript.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Just to note that you can also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation as well. Okay, I would now like to welcome our speaker for today's webinar, Kamil Sekulski, PhD, Field Application Scientist, GenScript. Kamil, I will go ahead and hand it over to you. You may now begin your presentation.
Thank you, Sydney. I will share my presentation right now. Hello, everyone. My name is Kamil Sekulski, and I'm Field Application Scientist at GenScript, and I'm excited to present you a GenScript automated one-stop purification solution for plasmid, antibody, and protein. And here's what I will cover today. I will start by setting the stage with a quick overview of the end-to-end processes from plasmid to protein. Next, I will give a quick introduction about our automated solutions for plasmid purification. But the majority of today's presentation will focus on protein purification. I will start by giving a brief overview of history and different methods and technologies, how they compare, and also how GenScript utilized one of these technologies to develop fully automated and high throughput mini and medium scale protein purification instruments. I will go over more details about them and also show you case studies. First, I would like to show you this typical laboratory workflow to obtain protein sample that I'm sure everyone here is familiar with. It's a multi-step process starting with the gene cloning, followed by plasmid purification, transformation, and transfections, protein expression, harvest, and finally protein purification. As we all know, it's crucial to achieve optimal yields and purity at every step to guarantee downstream success. And this is why having reliable tools is so important. GenScript provides tools for each of this step, streamlining the entire process. But today I will focus mostly on two of them, which is plasmid purification and mainly protein purification. When it comes to the automated solutions for plasmid purification GenScript offers, we offer two instruments. The first one is AmMag Quatro Maxi 1400, and another one for Maxi prep purifications, and another one is AmMag Quatro Mini 1100 for mini prep purifications. Those are high throughput instruments with a modular design, meaning they have controller, and you can connect up to 4 modules to that controller that run completely independently, and you can use different protocols for each of them. When it comes to Maxi Prep, one module can purify up to 6 samples at a time. So having all of them, you can purify around 24 samples, up to 24 samples in roughly 2.5 hours. The similar system applies to Mini. You can connect up to 4 modules with one, being able to process up to 48 samples. This is a higher scale, which gives us up to almost 200 samples, which you can also roughly purify in 2.5 hours at a time if you have all the modules. It also comes with the all-inclusive consumable kit, so all the kits and reagents are provided in the kit. There is also very easy to use interface that we built. And we also provide validated protocols that we've established, and most of the users actually use them. But if needed, we offer help to customize the protocols as well. Both instruments provide transfection-grade purified plasmid, And there are a few types of kits that we offer. There's regular kit, and there's also endo-free kit that's already available for the Maxi prep instrument. And the one for Mini is coming on soon. We're also about to launch the animal-free kit as well. I will not go over more details about those plasmid purification solutions, but I'll highly recommend you watch the previous webinar, which was fully dedicated to those instruments, and also any other materials you can find on the internet and GenScript website. And if you're interested and have any questions about them, feel free to reach out to us. What I would like to focus mainly today is protein purification. And here's just a brief overview of the history of it, which is really long and reaches 19th century, but it wasn't until mid-1916 when the affinity purification was introduced, which significantly improved selectivity. Then recombinant expression made scalable purification more accessible. And in the 21st century, the need for automation made FPL systems widely adopted, and those are still being used until now. So as you can see, over time, methods and systems have developed to support different needs, such as purity, scalability, and automation. And our current laboratory workflows definitely require all of them. But in the current fast-paced environment, another requirement has emerged. And it's a requirement for high throughput. And that's why GenScript developed this fully automated high throughput instrument that is based on the magnetic beads technology. But before I give you more details about the instrument itself, I want to explain you how and why the magnetic beads technology allowed us to fully automate the purification process. This slide shows the workflow for both of the techniques, magnetic beads and the resin columns that are the gold standard technique people use right now. And I'm sure you're all familiar with the workflow here. It starts with a few manual steps, such as cell lysis, unless it's a secreted protein, and clarification, which is definitely required to avoid clogging the columns. So typically centrifugation and filtration is being performed. And the first two steps, depending on the throughput and number of samples, can take even up to a few hours if we deal with multiple samples. After that, the instrument automates the sample loading, washes, and elutions. So when we thought at GenScript about developing automated instrument, we thought about full automation, reducing the manual steps to the minimum. And we found this bottleneck of the first two steps taking lots of time, hands-on time. And we wanted to either fully automate them or skip them if possible. And that's why we decided to integrate the magnetic beads solution, because with that method, we can fully automate the cell lysis on the instrument, and we can also skip the clarification, because here there is no column that can be clogged, and this doesn't affect the magnetic beads performance. And obviously, I will show you the data that supports that. The results you get with and without clarification with the magnetic beads are identical. And with that, we were able to automate all of those steps, significantly reducing the purification time. Here is a little bit more detailed workflow of I just mentioned and how it applies on our instruments. We have two instruments, as I mentioned, for mini and midi scale. And those workflows are almost identical when it comes to the instruments, but these slightly vary depending on the protein type. For cytosolic proteins and secreted proteins, those workflows are slightly different. So for mini scale, we typically grow our cells and express protein in plates. It can be 24, 48, or 96 well plate. We grow them, and then we have to centrifuge the cells and remove the media. And having the pellet in the plate, we just simply place it in the instrument, set it up, with the buffers and consumables, and the instrument automates all the steps, such as pre-suspension, lysis, incubation with beads, washes, and elution. So at the end of the run, you only come and you get your purified protein. If it's a secreted protein or antibody, for example, the workflow is even easier because you simply express it and you place it in the instrument. There is no manual steps required in between. There is no tricks here. We simply place it on the instrument, and then the instrument performs incubation with beads, washes, elutions, and also sometimes neutralization, especially if you use our antibody purification beads that elute in an acidic environment. The instrument can also perform neutralization. So applying this workflow, as you can see, this shortens the processing time quite significantly. With that, I would like to start with the introduction of the first protein purification instrument. It's the AmMag Quatro Mini 1100 for the mini scale. And you've already seen this instrument 3 slides ago, because this is exactly the same instrument that is used for the mini scale plasmid application, as it has a dual function. We use the same consumables, and the entire setup is very similar for both applications. The main difference is the reagents for plasmid we supply. For the protein purification, we don't supply buffers, as there's really no universal buffers for protein purification, so these needs to be made separately. And another difference is the throughput. I mentioned that for plasmid, one module can purify up to 48 samples. For protein, it's 96. A single module can purify up to 96 due to the module design, the instrument design. So a few key features for protein application. It's fully automated, as I just showed you the workflow. For secreted sample, it's from the cell culture, just directly goes to the protein. The instrument does everything for us. And for intracellular, we have to centrifuge to obtain the bacterial pellet. And from that point, the instrument will take care of the entire process. And there is no manual operation required during the run once we start it. When it comes to throughput for protein, as I mentioned, 96 samples in a single module. If you have all 4 of them connected to the single controller, you can process almost 400 samples at the same time. It's also compatible with 24, 48, or 96 well plates as an initial plate with your sample. It will depend on your throughput needs and also scale. The purification time adds around 30-60 minutes without the incubation. So obviously, you decide the incubation time, but typically the entire process, depending on the number of samples, can take around 2.5 hours. And the final sample is a protein with typically more than 90% purity sample, just from a single step purification. Here is just another representation of the instrument inside. It's a layout of the instrument from like a top view, highlighting different positions and also consumables. With number 1 is our plate with bacterial pellets or cell culture. This is where we insert it. Number 2 is buffer tanks. This is where we place our buffers. Number 3 is tip box. And number 4, we have reaction vessels. This is the part of the instrument where our sample spends the most time. This is where the binding with the beads occurs, as well as washes and elutions. Number 5, we have the magnetic beads reservoir. So this is a really useful option, so we don't have to add beads individually to every sample. We just add all of them to the reservoir, and then instrument adds them individually to every sample for us. Position 6 is our elution plate. Position 7 is a liquid waste box, and and right next to it there is also tip waste box. As I mentioned, all consumables come with our consumable kit. And now I would like to play a quick video, basically visualizing everything I just mentioned. This video was made specifically for the plasmid purification, as you will see from the description. However, the entire setup is very similar. The only difference, as I mentioned, is that we don't supply reagents for protein purification like we do for plasmid. And also the beads will be different for protein purification. All right. And with that, as I described the instrument, how it works, now I would like to show you a little bit of case studies to show you how the instrument actually performs. So the first data I would like to show you is, I mentioned a lot how we automated the cell lysis. And I would like to show you that, how we develop it. So we've spent a lot of time developing these protocols, and here's just one of the data I want to show you. For that specific experiment, we used histacked GST protein from E. coli strain. So this is for the bacterial lysis. The sample volume was 1 ml, and we used 25 microliters of settled beads, nickel charged MAG beads from GenScript per sample. And we've optimized it to use enzymatic lysis with lysosome, chemical lysis with detergents, and also we try to combine both. We've also used in this experiment some negative control, which was basically no lysis at all. As you can see from these results, they're comparable and all of them work. We typically notice that chemical lysis or both perform best, or at least for the shorter lysis period. You can also access the quality from the graph here and from the gel. Of course, for no lysis, there was no protein, and from enzymatic, chemical, or both, you can see how the protein looks on the gel. So obviously, we've developed those protocols, and if anyone is interested in our instrument, we also supply those protocols as reference as well. Having those said, we've been using them since that. And then I would like to show you another data that's actually from external user, who also purify histack protein, but compared how it performs, how Quatro 1100 compares with manual purification using beads. And the data, the performance you can see in the table when it comes to yield, or actually concentration here, you can see that for Quatro 1100 and manual purification, the yield was really comparable, and the same, they concluded similar purification results from instrument and manual. And the quality you can access on the gel from the Quatro 1100 and the manual, it looks very comparable. They even concluded that it's cleaner from the instrument. So the results you get really similar from that of manual purification, but obviously the instrument saves lots of time, especially if you do multiple samples like 96. I've also mentioned before that the instrument is compatible with 3 different plates, 24, 48, and the 96. And I just wanted to quickly show you how it performs and why you might want to choose one or the other in certain scenarios. So this specific experiment also was used histack GST in the E. coli strain. And obviously for those samples, the sample volume varies. So those are the maximum volumes that we use. For 96 well plate, it's 1 ml. For 48 well plate, it's 2 ml, and for 24 well plate, it's 4 ml. We use the same volume of beads for each of them. And the results you can see here for the yield, obviously the higher the sample volume with the same protein type, the higher the yield you can obtain. And that you can clearly see here that 24 well plate, since it was 4 ml, we got higher yield than, for example, 48 or 96. So depending on your skill and depending on your throughput, you have this flexibility to use either of those plate types. You can also see the consistency of the results on this graph. The results for the beads purification, especially if the instrument automated, is really good. The results are typically really consistent and also of a high quality, which again, I showed the gel that you can analyze. Another data I would like to show is that, so far I showed you the purification using nickel charged beads and histag protein. Here I want to show you something different, and it's the antibody purification of specifically human IgG from CHO cells. The sample volume was 1 ml, and for this we use 50 microliters of settled AmMag protein A beads, also from GenScript. Those are very popular beads and well-performing. The recovery rate was 92% and purity over 90% that you can analyze on the gel. As you can see, all nice clean protein. And the same when it comes to results. The yield or the concentration was plotted here. And of high consistency. You can see that CV of 3% suggests highly consistent results as well. So I've just showed you two types of protein that we purified, or I just showed you. But to be honest, you can use those instruments to purify any type of proteins as long as you have the magnetic beads that can support that. And at the end of my presentation, I have some table summary of the beads that GenScript offers, and I will also go over that. Now, I would like to scale it up a little bit and introduce you a new instrument that you haven't seen yet for the MIDI scale high throughput purification, and it's the AmMag Quatro ProAb 1300. It's a slightly different instrument that the design I will explain in the next slide. But the key features about this instrument is that it's fully automated as well, similar to the previous one. It can do lysis, incubation with beads, and all the purification steps like washes, elution, and neutralization as well. Again, no manual operation required once we press the start. When it comes to throughput, since it's a higher scale, the throughput is slightly smaller. It can purify 12 samples in a single run. The range of the subtle magnetic beads you can use per sample is between 100 microliters to up to 6 ml. So the range is really high. And with 6 ml of sample, obviously it will depend on the binding capacity of specific beads you use. But for some of them, you can purify even up to 240 milligram. The volume range it works within is between 1 and 65 ml. It saves time. The purification can take around 30 minutes if we exclude incubation. Obviously, we should include incubation as well, but then it's up to us how long we want to incubate it for. The workflow is very similar to the previous instrument I showed you, with one exception, or even improvement. When it comes to secreted proteins or antibodies, on the mini instrument, we only are able to incubate the beads in the instrument. For this instrument, since we typically grow in flask, we usually recommend actually adding beads to the culture the evening before purification. So the beads pre-incubate overnight with our sample, and then all we have to do, we have to just simply place it in the instrument and run the purification process without the incubation. And with that, the time is actually around 30-40 minutes. We also have flexibility to customize the protocols, and we have many steps that we can modify. And I will explain that in a moment. This is how the instrument looks like from the inside. I mentioned that it looks different. First, the controller is different. It's actually attached to each instrument, and it's in the form of tablet, as you can see. There is this turntable with 6 different stations that we place our tubes with sample, and this rotates. We also have a mixing station and dispensing station at the back that you will see a little bit better in the next slides and also video that I will show you. And another useful function that actually all the instruments have, I didn't mention that before, is the UV lamp. So this allows us to decontaminate the inside of the instrument after every run if we want to. Here is a little bit different representation of this instrument. This is like a top view of this carousel I showed you with those different stations. I mentioned there are 6 stations. One is at exposition. It's for the sample where we input our sample. And then we have 5 extra, A, B, C, D, and E, that we can customize however we want. So in this specific example on the slide, it's customized to have wash 1, wash 2, wash 3, and then elution 1 and elution 2. However, it's really up to you how you want to customize it. You can do 2 washers and 3 elutions. You don't have to use all those positions. You can do even 1 wash, 1 elution. It's really up to you. And we're actually not even limited to 5 steps anymore. We just recently introduced a new software that enables us to customize up to 8 steps. So this will be possible too. A little bit closer view at those stations and consumables. So number 1 is the dispensing workstation. The way it works, we prepare our buffers in bottles. And so we don't have to dispense them individually to every tube. The instrument will do it for us with the needles you can see here. We just have to program it in our method. Number 2 is the magnetic rod array with the sleeves. So this magnetic rod is what goes into the tube to attract the beads, capture the beads, and then goes up. The carousel rotates by 1 position, and it goes down and releases the beads in the next station. It never directly touches the liquid or any sample, so it allows for very easy maintenance of the instrument because it goes into those sleeves. This is like a plastic rod sleeve. It's a consumable. So this is actually what touches the sample, and then we just trash it. We also supply 100 ml, 50 ml, and also 25 ml, which are not shown here, tubes to allow you for different volumes and different purifications. It comes with really user-friendly interface, which actually is similar across all our instruments, but I wanted to show you here. So once we have our method saved, from that point, everything is really simple, and it just requires few steps. We have to first create the task where we select our program, number of samples, and also the magnetic beads volume that we will use. After this window will pop up that has a really useful function that calculates the buffer volumes. So we don't have to do math in our head of how much buffers to prepare. It will all calculate for us based on the method and number of samples. After we have everything connected and set, we just click start, and everything from that point will be automated. And again, I have a video that I want to play you that visualizes most of what I just told you about.
With just one click, Quatro, usherings in the era of automation. Say goodbye to tedious experimental steps. Simply put your samples into Quatro. It takes care of automated incubation, liquid handling, and transfer. You don't have to be limited by the sample volume of automatic purification instruments. Quatro single tube accommodates sample volumes of up to 65 milliliters, offering flexible solutions to meet your purification needs. Say goodbye to low productivity. In just 30 minutes, Quatro guides you effortlessly from cell supernatant to pure protein. The AmMag Quatro is your expert in automated purification.
All right, and now I will move to case studies on the AmMag Quatro 1300. So similarly to Mini, I wanted to just quickly show you the cell lysis protocols that we introduced here as well. It works in a similar fashion, and we use similar methodology, like GST, histag GST protein, E. coli. We just use bigger cell palette, obviously, and more beads, 500 microliters of settled AmMag nickel charge beads. As I mentioned, the range is from 100 microliters up to 6 ml. This is also an example of the protocol method that you customize. It's really simple, and we also provide trainings on how to do that. So it's really easy and straightforward to use. The results are shown here, and as you can see, we've tried enzymatic, chemical, and both. And we also compared to the physical lysis, which was sonication in this point, to see how it performs. And you can see that both chemical or enzymatic and chemical achieve similar results to the one of physical. That way, we don't have to do it manually anymore. We can simply let instruments do it for us. Here is also the example of the gel with the elutions that we achieved. We also compared how the instrument and the beads purification compares with resin. For that, we purify antibody, and we used our protein A beads, which I also mentioned before, and compared with the monoaffinity A resin and on the gravity column. The results you can see here in the table, and also here on the graph. You can see that beads outperform resin, even adjusting for the binding capacity. And the quality, you can see the purity, you can see on the gel. The non-reducing is here for the Quatro and for column, and also the reducing gel for the samples is here Quatro and here columns. That you can see that purity is very similar, even by eye, without quantifying it. But the most important aspect here is the results are more consistent using beads and definitely using the instrument. The CV is around 1.6 for beads and almost 12 for the resin. Not to mention how much faster it is to use the instrument. And another aspect I want to touch on, I have two case studies from external users, separate external users. I've mentioned how we integrated lysis on the instrument. And at the beginning, I also mentioned that we are able to skip the clarification process completely, right? But I haven't showed you the data yet. So here we go. This is the data from one of the users for antibody purification. And what was done here, two proteins were purified, protein 1 and protein 2, and using either filtration with 0.2 micron membrane filter or without filtration. The same for the second protein with filtration and without filtration. And there was one extra sample added as a control with gravity column to see how it compares. And if you quickly look at the gel, I think the results are pretty self-explanatory. And their conclusion was similar yield, even though they never showed us yield, but similar yield and purity was achieved. And I think that this gel nicely visualizes this conclusion. And another external user data, independent from the previous one, performed very similar experiment with crude and clear lysate, but this time on the histag protein. And the data, the results you can see in the table, they also used two proteins, target 1 and target 2. Crude lysate and clear lysate, if you compare the same here, crude lysate and clear lysate, you can see that the results are very comparable. And also the purity on the gel for the crude lysate and the clear lysate are really comparable. The same was their conclusion. Similar purification results from crude and clear lysate. And this just supports the data that we got internally as well, which I'm not showing because I prefer to show the external user data. So to summarize the comparison between the traditional method and the automated instrument for the MIDI scale, the ProAb 1300, we can highlight a few features. The first one is the hands-on time. That's, as I mentioned, our workflow allows us to spend much less time on the manual steps with our instrument. It also allows us for high reproducibility, which I also highlighted. The purification is much faster since we can skip those few manual steps or they're automated. The throughput is higher. We can purify up to 12 samples in a single module. For these traditional methods, we typically can do 1 sample at a time. There's also lower sample loss. And what's actually might be the key point sometimes is the skills required to operate it. We try to design an instrument to reduce that doesn't require much of the learning curve. So all we have to do is just set it up and click start. So the skills requirements are much lower for that instrument. And finally, I want to compare the two instruments I showed you, Quatro Mini 1100 and 1400. They're both useful and they're both for different needs and different applications. The first one is mostly for the hit screening since it's a high throughput, but of much lower capacity. So you can get up to 4 milligrams of protein per sample. So it's perfect for screening proteins. However, Quatro ProAb 1300 is of a larger scale, but slightly lower throughput. So this is best for the larger scale purification applications. And I also mentioned that you can purify any type of proteins as long as there are magnetic beads available. And that's true. And I wanted to show you some of the ones that GenScript offers. This table just highlights beads for antibody purifications. So we offer many different protein A beads, protein G beads that are more specific towards like mouse IgG. We also have a mix of protein A and G beads. We also have protein L that are more specific towards the light chain of the IgG, which I just noticed it's not in the table. And we also have the anti-VHH beads as well as some affinity purification beads, such as nickel charge beads, glutathione, streptavidin. We're also developing streptac beads right now, and actually many others. So if even something is not on the list here, and you're interested, feel free to reach out to us, and there is a chance that it's the beads that we're actually developing right now. So to conclude, GenScript offers tools for each of the steps from plasmid to protein and antibody purification. Not only the one I just mentioned, but also for the PCR reagents, cloning reagents such as GenBuilder and endonuclease. But the most popular tools from GenScript are definitely the Quatro instrument, all the one I showed you, Quatro Mini, ProAb, and also Quatro Maxi. With that, I would like to thank you for listening, and I'll be happy to take any questions you may have. Thank you.
All right, perfect. Thank you, Kamil, for the very informative presentation. We will now go ahead and move into the live Q&A portion of the webinar. So if you happen to have a question that you would like to ask, please go ahead and do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. All right, let's go ahead and jump in here. The first question that we have here says, do you offer demos for these instruments?
Yes, we offer demos for the instruments. There are a few types of demos we offer, and this is case-by-case scenario. We typically start with the virtual demo where we show the instrument virtually. Sometimes we also process customer sample if they're willing to send to us. And then we also offer different types of on-site demos, like demo and demo to buy options. So if you're ever interested in any of those demos, please reach out to us. We will set up a meeting and we'll try to discuss the best option for you.
Perfect. The next one that we have here says, how long does the purification take?
Yes, this will highly depend on the type of sample you want to purify and number of samples. Of course, number of samples is self-explanatory. The fewer samples you have, the faster it will be. With the type of samples, as I mentioned, if it's a secreted protein, the purification might take faster because it requires fewer steps. For intracellular proteins, it will take a little bit longer. But typically, the purifications take any time between like 30 minutes for ProAb, for antibodies, if it was pre-incubated, up to like 2.5 hours for like mini and for longer incubations.
Okay, wonderful. The last question that we have here says, do you provide training or tech support for these instruments?
Yes, we provide both. We provide training. If you're interested in buying or demoing the instrument, we'll either me or one of our FAEs in the group will go on site and provide you detailed training. So you don't have to guess what to do, even though it's all easy. But we will describe everything and show you every single step and answer any technical questions you may have at the time. And then even after training, we're still easy to reach for any technical support that we definitely provide.
Perfect. Okay. Thank you so much. And just wanted to thank you again for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, GenScript, for underwriting today's educational webinar. Before we go today, I would like to thank the audience for joining us and for their interesting questions. Questions that we did not have time for and those submitted during the on-demand period will be addressed by the speaker via the contact info you provided at the time of registration. As a reminder, this webinar can be viewed on demand. LabRoots will go ahead and alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. All right, until next time, thanks so much, everybody. Bye-bye.
Thank you.